It's either way is fun. I can wait. There are still many people outside. Oh, that's wait. Yeah. No, I'm okay. Yeah, I already had some water before I came here.
I think we will start now. And today's speaker is Jing Li. And Jing has received his Ph.D. from California's in computer science from University of California, Riverside in 2004. And he has been a faculty member in EECS department since then. And his research is in computational biology and statistical genetics and bioinformatics. And he will be talking about computational challenges in the era of personal genomics today, a subjective view. Okay, Jing.
Thank you very much, Mara. It's a great pleasure to talk my own research again in our home department. And thank you very much for coming. Okay. So today's talk is actually going to be a very high-level talk. Essentially, I assume you do not have any knowledge about computational biology bioinformatics. And I want to tell you what I'm doing, basically. Okay. So that's why it's called a subjective view. I'm going to first talk a little bit about what do I mean by personal genomics. Okay. So I think many of us already know something about the Human Genome Project. I think it's essentially the start of the whole field. Many of us also have seen these two covers from the top two journals, one is Science and Nature. Okay. So it's around 2001. These two top journals actually devote a whole issue to the project, the result from this Human Genome Project. One is from the public domain, another is from private sector. Okay. So briefly, what is a Human Genome Project? What do we get from this Human Genome Project? This is actually an international collaboration from a couple of countries. It first started from in 1990, and the original plan is a 15 years project, a collaboration from a few countries to figure out the DNA sequence of humans. Okay, the first draft was published in 2001, and later on in 2003, they declared they finished the whole sequence. In terms of cost, it's roughly about $3 billion. This is not a sequence only, the overall cost. Okay, so how do you view this number? Well, it is a big number in terms, if you are talking about personal genomics. I don't think anybody would like to spend $3 billion to figure out what is your DNA sequence. Okay. On the other hand, it's a small number compared to the $700 billion buyout plan for the Wall Street. Okay. It's only 0.5% of that money. Okay. What do we get out from this investment? Well, first, it's a reference sequence. Direct result from this project is the 3 billion base pairs of a DNA sequence. Definitely, we want to make sense of this data. Then there are many developments in terms of how to recognize the gene structure of this sequence. Well, there are some predictions. For example, how many genes are there? What are they? What are their structures? Okay. So roughly, let's say, originally before the genome is available, people estimate that maybe we have 10,000 genes. And later on, this number reduced to 5,000, 3,000. Now the consensus is about 20 to 25,000 genes. We still don't know exactly how many of them. And also, when we talk about genes, now we are talking about something called protein coding genes. Okay, there are some other things going on, but I'm not going to go to that details. But these many genes only account for a small fraction of that 3 billion base pairs. Base pairs means ATGC sequence, DNA sequences. Okay. That's some direct result from this project. But in addition to this, actually, we gain a lot of other things. In terms of the promote of this whole field, genomics, bioinformatics, computational biology, and all other kind of omics. You're going to see all those terms many times. And also in terms of technology, definitely sequencing technology, and all other related high throughput technologies. For example, microarray data technology for expression level, and many other high throughput technologies. And also, this has a greater impact on the society. For example, if one day, I think in the near future, every of us will have our own DNA sequence available in some chips, for example, then people can take advantage of that information. For example, when you buy life insurance, they want to take a look at your gene makeup to see whether you have some high risk of many different diseases. And then we have to think about these consequences. Therefore, earlier this year, actually, Congress passed some law, non-discrimination of genetic information. These are all the indirect consequences of this big project, in my view. Then for our students, people in computer science, EECS, what does this mean? This basically, we can say, we now have our source code for some program, okay? But it was written in some languages we don't know the grammar. So we want to make the sense of what does this mean to us, okay? Finally, after we finish this human genome project, is there anything else we can do? Or shall we do some more? Actually, yes. The answer definitely is yes. We are not going to stop those sequencer machines. Then the question is directly related to whose genome is this? It's called a human genome. It's for all human. On the other hand, it's nobody's genome. It's a reference sequence. It's a consensus thing. In addition to that, it's actually not a complete copy of genome. It's only about half of the copy. If you recall from your high school biological genetic textbook, we all have 23 pairs of chromosomes, illustrated over here. And these 3 billions actually is only half of these chromosomes. Each pair, we know one is from our father, another is from mother. This is only one of these 2 pairs, one of these 2 chromosomes. This is half of our genome, actually. Okay. Then, does this another half matter? It definitely matters, right? Then why we only obtain this half of them? It's because essentially at that time it's a limitation, let's say, technology. Okay. So this is a portion of a gene, okay, one important gene, just to illustrate what it looks like. Okay. Then, around early 2002, there's another very important international project, collaborations. It's called the HapMap project. So what is the goal of HapMap project? It's actually, the goal is we want to know more about individual differences in terms of their DNA sequences. And we want to know actually eventually how these differences corresponding to the differences of our appearance, our risks to disease. Okay. So at that time, people decide, okay, let's go ahead to find about 270 individuals from different populations, like white, black, Chinese, Japanese. Okay. Let's take a look at what are the differences among this group of individuals. Okay. That's the major goal of this international HapMap project. The raw data was collected in 3 years, and the analysis is still ongoing. And recently, they published the phase 3 data release. Okay. So, what is exactly the? Okay, the cost is about 100 million. Okay, for this 270 individuals. I said this might have a direct consequence on health care. Okay, why is that? Look at this picture. What they did is, they didn't do the sequencing for all these 270 individuals. Instead, they only look at the differences. For example, over here we have 4 chromosomes from 4 different individuals. Okay, you look at these chromosomes. Actually, you look at the sequences. Most of the places they are the same. It's obvious. We are human. Okay. On the other hand, there are some places like these places. They may have some differences. This difference is only one single nucleotide change. It's C in these 3 individuals, but there's a T in this individual. We call this SNP, or single nucleotide polymorphisms. Okay. So the goal of this project is basically first, we want to know where these differences and how can we measure these differences. Okay. Once we have these differences, we can ignore all these common things, and then we're going to have something we call the haplotype. This is the one individual corresponding to chromosome 1, but we only record those differences. It's over here. The 3 positions over here. Position 1 over here, position 2 over here, and position 3 over here. You consider this 3 billions, you're going to see a lot of those things. Okay, we call this haplotype. We're going to mention again later on. So again, this haplotype is only one of the 2 chromosomes, 2 homolog chromosomes, the 2 chromosomes from our parents. Okay. Another goal of this project is to take a look at the correlations of these differences, because definitely they are not independent. Okay, and then there are many problems, because when we measure these differences, we measure something called genotype. That is the two letters on the pair of chromosomes. But when we talk about the haplotype, it's actually the differences on a single chromosome. So they are definitely going to have some computational problem. That is, from your measurement, how can you infer those kind of haplotypes? That is, how do you know the correlations of these differences on each of the chromosomes, on each pair of them? In addition to that problem, there's another problem which is quite hard in a couple of years ago. That is, we have many of those differences. If they are correlated, we do not need to know every of them. A subset of them will give us enough information with a lower cost. Therefore, say currently we know about couple million of these differences. But can we actually get a couple thousand, 100,000 of these SNPs to represent all those variations? That problem is called a tag SNP selection problem. So how you computationally select, say, a minimum number of a subset of these differences to represent the whole variation. Okay. That's what we have observed in past few years. And how about in the future? What do I mean by personal genomics? Actually, there are a couple of things. After the HapMap project, there's another project. It's called 1,000 genomes. The purpose of this project is let's collect 1,000 individuals. Let's get their sequences other than these differences. Because SNPs is only one kind of variation. There are many other kind of variations. The HapMap project did not capture. Okay. If you obtain 1,000 individuals, the whole genome of them, and then you can have a better idea what other genetic variations. Something later on I'm going to mention, like a copy number variations. Different segment, you have multiple occurrence. Okay. Now the cost was estimated to get one genome from each individual. You might actually need to spend $300,000 to $1 million. This is greatly reduced already from 3 billion, right? If you're only interested in those differences instead of the sequence, you can actually spend less than $1,000 to obtain all those SNPs, not all those SNPs, those tag SNPs. There are many companies actually doing this right now. For example, one, you all know Google, but actually this company has a close tie with Google. It gets some money from Google, and you can obtain all those SNPs differences from this company. But again, this one you see it's still at the SNP level. It's not at the sequence level. But in a few years, people are talking about $1,000 genome. That is really, we are going to get every letter from our DNA sequence with cost of less than $1,000. This is the estimate, okay? But probably it might be shorter than this time. Then we really in the area of personal genomics. Each one maybe, because this is static information, it's a once a time life event. You can test with $1,000. It's not going to change in your life. Okay, then definitely we are not ready computationally to address these problems. Okay, you think about the genetic study. First, the people find some very small number of markers. They want to find an association of markers and phenotypic differences. Now you have the whole genome. What are you going to do? The more the better, or the less the better? We're really not ready to address this issue yet. So finally, for personal genomics, it's all about me. It's about my gene. It's about my risk. Okay. So when we talk about the high throughput technology for personal genomics, there's going to be a huge challenge. Data management, data analysis, because raw data does not make sense. We want to make some inference knowledge from that. Okay. And hopefully that knowledge will help us to promote our health. Okay. Given the change in the collecting data, the fundamental issue is still the same. Essentially, the relationship between genes and disease. Sometimes they call it genotypes and phenotypes. Okay. Which gene actually might increase in what level of particular disease? Okay. This problem has been long been known in genetics. People always investigate this problem. But the whole picture now actually changed. You can essentially know every piece of variation in your genetic material. And then can you do better in tackle this relationship? We don't know, actually. Then for computer science people, you can think about, oh, now I have some source code written in some language we don't know mostly. And then we actually want to predict its running behaviors. Okay, you also have different versions of this source code, and you don't understand what the language is, and you observe some behaviors of your program. You try to see their links. Okay, and someday we hope that we might actually rewrite some of those codes. Okay, so that's a brief introduction. What do I mean by personal genomics? It's really, we already opened the door to the personal genomics area. And then I'm going to talk about my own research that is closely related to this concept. Okay, so I have a couple of ongoing research projects. And some are future directions. Even though you see there are so many, actually some of them are quite related. Okay, so I'm going to go over this at a very high level, and if you have questions, details, we can talk about more. Okay. The first one is association test. We are talking about the problem of how can you establish a relationship between genes and phenotypes and disease. Okay, one way to do that is very simple. It's called association test, association analysis. Actually, everybody knows this kind of test. For example, let's go back to this famous example. We want to know whether smoking and lung cancer have any relationship or not. Then it's very easy. You collect, say, 100 people who have lung cancer, 100 people who don't. And then you look at, among these 100, how many of them are smoking, how many of them do not smoke. And then you're going to have a solution like this, a condition table. Okay. Based on this table, you can construct some statistical tests to see whether these two factors, they are independent or not. Simple as like this. Then, how about a genetic test? What you did is basically, let's change this condition to those differences. Instead of non-smoking, we are talking about A or T.
One position?
Yes, that's an excellent question. Yeah, we can do this. The problem is, oh, okay, next slide. I'll skip this slide, go to your question. The problem is, we have 500,000 of these SNPs. If you test one by one and you set some threshold significance, by default, you're going to have many false positives. This is in statistics we call something called a multiple testing problem. Okay. In addition to that, there are many other issues I'm going to briefly mention. Okay. But let's go back one step. So basically, how can association tell us position information? This is something related the concept called linked disequilibrium. But you don't need to bother with that. It's basically some correlation. So this will illustrate how can we use association information to predict position. Suppose in the past we have some ancestral haplotypes. When we talk about the haplotypes, because there are correlations, you're going to see segment of haplotypes among population, they have some kind of frequencies. You're going to see different people might share the same segment of haplotypes. Therefore, you're going to have the count, you're going to have the frequencies. And then suppose at some point, there's a mutation occur over here, which is going to increase the risk of some disease. Okay? And then you look at the haplotype structure at the moment, the current state, current days. Then you might expect something like this. This means each of these haplotypes actually is a combination of previous ancestral haplotypes. It's mainly, there are two forces over here. One is something called recombination, another is called mutation. Why we have this block? Because when people inherit a chromosome from parent, they inherit a big chunk of chromosome, not only single pair by single pair. Okay. After many generations, because of recombination, you're going to see many different boundaries between blocks. Okay. Over here, I did not illustrate mutations, but we do have mutations. Then you collect a group of people who are affected. You collect a group of people who are normal with respect to a particular disease, and then you want to figure out where this mutation occurs. If I don't have this, you directly look at this, it's hard to see. But if I tell you it is actually over here, then you're going to see some patterns. What patterns? You see, this group of people who are affected, they all share a segment of this common haplotype where the mutation occurs. But however, this sharing actually is quite a noise. You know, this one is from here to here. This one from here to here. The length and the position are all different. On top of that, they also have mutations. But you don't see sharing in these normal individuals. Then you apply some kind of test to test this association. If you see some strong associations at some position, you're going to suspect that might be due to this reason. That's why association can give some information about the position. Okay. This is the underlying idea why we can do gene prediction, gene position prediction. Now let's face the problem. One problem is we have too many of these markers. In addition to that, there are other problems, like something we call the nearby SNPs. They are correlated. This you do this independent test, it does not capture this, and the result can be very noisy. Because even though the two markers are very close to each other, they do not give consistent result about the association. Okay. Another thing, the real causal mutation, you might not be able to type them. You might not be able to measure them. Okay. In addition to that, there are many other issues. When we talk about disease, we are commonly talk about something called a complex disease, which means there are many genes that contribute to the risk of disease. And when you see a patient, it does not necessarily mean that patient is going to have that mutation. When you see a normal people, it does not mean that normal people do not have that mutation that increase the risk. It's just time is not yet, has not developed that disease yet. So basically, you're going to have false positives and false negatives. Yeah. Another very important factor is something called gene-environment interaction. This is very easy to get. Whether you're going to be healthy or not, it not only depends on your gene, it also depends on your what you eat and what is your style doing exercise. All those kind of things. And many of those, it's very difficult to measure. Okay. So therefore, to tackle this gene and disease relationship is really a hard problem. And unfortunately, many of those common diseases we heard about are those complex diseases. Okay. So one alternative a lot of people are interested in is called, instead of those single SNP tests, people talk about something called a Haplotype-based method. That is, you consider a segment of SNPs, simultaneously. Okay. Why this might work? This is a cartoon talk example to show why this might be better than single SNP test. For example, we have two positions. Over here, I use 1, 2 to denote those ATGCs because it doesn't matter. And then you look at their distribution in 100 normal individuals, 100 patients. You might see something like this. That is, this haplotype, the distribution, 50 cases has this haplotype, 10 controls has this haplotype, and so on and so forth for all others. Then you look at them individually. You're going to see, okay, let's first take a look at this one. You look at how many ones occur in cases. There are 50 of them. Then how many twos, how many ones occur in the normal individual? You see, there's a 1, there's 40 of them, there's a 1, 10 of them. You see, the 1 occur in cases 50 times. It also occurs in controls, normal individual 50 times. And you look at the 2, it's also 50, and 10 plus 40, 50. Then you look at this locus 1 individually. You're going to see there are no relationship about this position and the disease. This is a small table we mentioned earlier. The same thing for locus 2. If you look at them the same, there are no correlations. But if we look at them together, definitely see these two haplotypes actually occur more often in cases than controls. These two actually occur more often in controls. Then you're going to gain something by looking at them jointly. Okay, so that's definitely just a toy example, but it tells us it may have some advantage by thinking about those haplotype segments when you do the association. Okay, what we did over here is basically we developed some algorithm and implemented into a software called HapMiner. This algorithm in the high level is very simple. You take the haplotype segment among controls and among patients, and then you look at their similarities. You group those similar haplotypes into one cluster. Then let's see among that cluster how many of them are cases, how many of them are controls. If you see an overrepresentation of cases in your cluster, you might suspect that segment of haplotype is related to the disease. Okay. But we have to deal with all those previous problems, noise. Therefore, we take something called density-based clustering. We can ignore at some level those noise. Okay. And we did some extensions based on this basic idea. For example, not only disease status, we can consider quantitative traits, height, weight. Those are all quantitative traits, but that is related to some quantitative traits related to disease. And we can handle something called haplotype ambiguity. I'm going to talk about this in a moment. What does that mean? And right now, we try to extend this to family data to include individuals within a family. Some other project is all related to this haplotype thing, because remember I mentioned earlier, haplotype is not directly observable. We only observe genotypes, that is a pair of alleles on a pair of chromosomes, but not a single chromosome. Then there's another problem, how actually you obtain those haplotypes. Okay. Again, this is one of the major goal for HapMap project. Why they call it HapMap? They want to infer the haplotype structure and haplotypes for different populations. This again, in a few years earlier, this is a very hot topic. What we did over here is actually we worked on the family-based haplotype inference problem. When you have family, this problem becomes easier because we all know something called Mendelian law, that if there are two alleles in a parent, a child actually only inherit exactly one from each of the parents. Okay, then you're going to have some constraints when you infer which one is from father, which one is from mother. Okay, so that okay. I'll first give you some example. Okay, so this is a family of 4 members, parents and the kids. Let's consider two positions. Okay, then we have some assumption that exactly one of these two is from each of these parents. Okay, then you see this one comes from this parent, this one comes from this parent. Okay, so there's some ambiguity over here, or freedom. Okay. And then the problem is how you define which assignment is the true assignment. See, I have 2 assignments over here. This assignment means that, for example, look at this position. This assignment means that this one is from this parent, this 2 is from this parent. On the other hand, this one assumes this 2 is from this parent, this one is from this parent. But which one is more likely the truth? Well, this relates to model selection, right? You can either define statistically, or you can either define combinatorially. For example, over here we take a combinatorial approach. We would like to find Okay. By looking at these 2 examples, we count the number of recombination. And we assume that the small number of recombination would be better. That is because when we look at those SNPs, they are very closely spaced. Therefore, the recombination is a rare event. Okay. In this case, what do I mean by recombination? You can take a look at this example. You see this kid. It inherited the 1-1 haplotype from this parent. Therefore, there are no recombination. But it also has a 1-2 haplotype. But this parent do not have a 1-2 haplotype. It only has 1-1 and 2-2. These 2 are 2 chromosomes. Therefore, there must be a crossover. Inherit this one to here and inherit this one to here. This kind of crossover we call recombination. We want to minimize this kind of recombination. Okay. And in the extreme case, we want to find all the solutions with the 0 recombination, no recombination. Okay. How are you gonna which problem, which definition of the problem you're gonna tackle? It depends on your data and also on your purpose. Because this recombination is closely related to the marker, the interval distance. If it is high, then you're going to see more recombination. If it is low, you're going to see fewer recombinations. Therefore, how you formulate this problem is really dependent on these 2 things. Okay. You see, this problem probably is easy for this example. You can essentially enumerate all the possible configurations and then count which one has the minimum one. But the problem is getting hard because you not only have this, but also have this kind of structure. Okay. This is a human pedigree structure. It has a couple of generations. Not only that, you only have data at those leaves, those shaded shapes. You have all those individuals, they do not have that genetic information. Then you see it's really hard to infer because these individuals are all related, but you miss all the information about their parents, their grandparents, and so on and so forth. This constraint, this one has some constraint on this one, but it's through a very long path in this graph. Then you see a lot of freedoms, and you figure out what exactly this constraint on this one. It is going to be the number of possibilities is exponentially large. Okay. Over here is another very complicated pedigree structure, but this one is an animal pedigree. Okay, it's not a human pedigree. Okay. So again, you have the same problem. Not only the pedigree structure is very complicated, you also have a lot of things are missed. Okay. To figure out exactly the haploid configuration of those individuals by taking in consideration all those constraints, it's going to be extremely hard. Okay. The three factors affect the complexity of this problem is essentially the pedigree size and structure, how big, how complicated, and the number of SNPs. That's definitely a factor, and also the missing read and patterns. Okay. For this problem, we have worked on this problem for a couple of years and developed some algorithm for it. While we have so many different algorithms, essentially we made some assumption to make the problem easier. But taking advantage of those assumptions, we can develop different algorithms for that special case. Okay. There are many related work on this specific problem too, in terms of complexity as well as algorithms. Okay. I will briefly mention one of the recent development by a student in our group. So this one tackles this problem. That is, if there are no recombination in this pedigree, how you actually get all the configurations with no recombination. Okay. The high-level idea of this algorithm is, well, let's first assume the pedigree is very simple. There are no loops, mating loops, and there are no missing data. In the ideal case, whether we can efficiently solve this problem. Okay. It turns out in this ideal case, yes, with some elegant structure, disjoint set, we can actually find all the solutions with no recombination. Okay. We change this problem using, we represent those constraints using some binary variables. And then we manipulate those binary variables using disjoint set structure. Essentially, those binary variables, the relationship becomes they are the same. They do not the same. They are not the same. Okay, and then we can represent the solution, nice way such that the representation, you can obtain the representation in linear, almost linear time. It even does not depend on how many solutions you have. Okay, that's something we call a general solution. It's a representation of all the possible configurations. Okay. There are some details. I'm going to skip this. And then once we have this algorithm, we extend this core algorithm to looped pedigrees, as well as pedigrees with missing data. Okay. And currently, we are still working on some other directions. For example, what if you have multiple pedigrees? Whether you should take into consideration the information from different pedigrees. Okay. And also, if you really have a large number of markers, that is across the whole genome, there's still a problem for this formulation. Because we are looking for solutions with zero recombination. But if you consider the whole genome, there are definitely some recombination. And there are actually many recombinations if the pedigree is large. Then you cannot directly apply this. Then how to extend to that? Okay. Over here, I'll show one slide of the result in terms of efficiency of this algorithm. We compare this with one. It's called RLP. It's our previous implementation of another algorithm we developed. Another is Merlin. It's a very popular statistical package. The new algorithm is denoted as DSS, disjoint set structure. You see, all those three algorithms are exact algorithms, meaning that they all give the same answer for an input. Okay. Therefore, we only need to look at their efficiency. we have done some experiments with respect to different number of SNPs you considered and different pedigree structures as well as different missing rate. Okay. You can see actually this algorithm is almost finished all this experiment in less than 0.1 second. These two algorithms, actually, both of them, theoretically, we know they are exponential algorithms.
What is the difference between DSS 17 and 29?
Oh, that's the size of the pedigree. 17 members, 29 members, 52, 128. Yeah, this is a log scale. You see, so you see the difference is obvious. Okay. That is because mainly we find some special relationship about those constraints on that graph. Okay. Okay. I have some other project. I'm gonna go over very quickly. For example, this one. You might have heard about the Genome-wide association study. It's a big thing at NIH. It's essentially the association test I said earlier. But association does not mean association itself does not mean there are some causal relationship between that gene and the disease. It's just independence of two variables. To verify those kind of associations, you have to do something called a replication test. Therefore, for genome-wide association study, usually people take an approach like this. You first have a group of cases and controls. You do the whole genome scan. From there, you pick about 20,000 SNPs that show significance. And then you recruit another group of individuals, only test on this many SNPs. And to use that to narrow down this to another number, and then do it again on another group of individuals. Okay. And finally, hopefully, you can find some candidate. Because you see, these two groups, they are independent. Therefore, if they are significant, those SNPs are significant from these two groups, that give you stronger evidence that those SNPs might be really interesting. Okay. Then the problem is, how are you going to pick a subset from here to here and to here to here? Okay. So we have done two things for that problem. One is we use some information measure to prioritize those subset of SNPs. Basically, we define something called minimum redundancy, maximum relevance with respect to the disease. And we try to identify those subsets. Another work along this line is when we try to identify the subset, we actually consider the interactions between different SNPs. Okay. But when we talk about interactions, then there's a big issue. Because you already have 500,000 of SNPs or variables. Then you talk about their interactions. Then you're going to have 500,000 to the power of whatever the degree of interaction you're going to consider. That's going to be a huge challenge. Even for two-way interaction, you have 500,000 to the power of 2. Okay. Then is it feasible actually to consider multiple interactions? We still don't know. People, at least at the moment, it's not, mostly it's not feasible. Okay. And now we are trying to use some decision tree and some essential, some boosting approach to see whether we can tackle this problem. Okay. I want to spend a few minutes on some biological findings after we apply those algorithms. This is about one special disease. It's called sporonic ALS. Okay. This is a neurological disease. Okay. We applied our approach on one data set, which is one of the first available genome-wide association data set. What we find is we find some genes and we look at their functionality annotation, and we see some of them involve cell cycles, and some of them involve ion binding, ion transporting. Whether this is meaningful or not, we cannot do wet lab experiments, but we can compare this with some existing knowledge. For example, it is known that this disease actually was caused by the degeneration and death of motor neurons. So this, from there, you see definitely something related to a cell cycle. Okay. In addition to that, we look at some other evidence for familiar ALS cases. People use other approach, like linked analysis, find some genes, it's called SOD1, is linked to this disease. This is different from there because this one you might observe a couple of affect individuals within one family, but this one you only observe one from a family. And you see there's some relationship of this gene and this gene because they all of them are ion binding. Okay. And this is a final result we obtained for those SNPs. Where are they and which gene are they within, and then their functionality. It seems that there are some interesting going on there. Okay, but we need some further analysis of this. Okay, we also did some expression analysis, but given time, I'm going to skip this. So basically, we looked at something called gene co-expression network. We want to find some signatures that across multiple species. Okay. Together with Dr. Mehmet Koyter, we're actually working on another project on the analysis of biological networks. Again, we want to use some evolutionary information to look at the networks from different species. Okay, some future work. Why is it related to haplotype structure? What does that mean? I said the haplotype is a key concept in this talk. We mentioned a lot of times. Haplotype, there are some correlations among those differences. This correlation is actually very different at different places. How can you capture those correlations? In this project, we try to use some graphic models, like this one. Like this one, to capture those correlations. These graphic models basically are generative model like a hidden macro model. The major difference of this with the hidden macro model is we actually consider, for hidden macro model, most cases you only consider first order. Okay. But over here, we actually consider multiple orders. But instead of direct extension, we model them as blocks. These blocks are controlled by some parameters over here, and within these blocks, they are going to generate some ancestral haplotypes, and for each ancestral haplotype, we might have some mutation with some parameter mutation rate, and then we're going to observe current haplotypes. As I said, you can only observe genotypes. This is not enough. Actually, we can pair two such models together to generate genotypes. That is basically what this figure illustrates. Okay. Another thing I mentioned is copy number variation. Copy number variation is another type of variation in addition to SNPs. Okay. This has been found in many cancer cells, which are not inheritable variation, but still people want to identify which part of the chromosome might have those kind of copy number variations. And these are the figure you're going to see for typical copy number variation data. You can see a lot of noise. You want to infer whether which part is different from the normal copies. And we worked on a few different models for this problem. Okay, RNA. there's something called a central dogma in molecular biology. We only consider about DNA. We also talk about RNA and protein. Okay. Recently, it has been shown that RNA actually is very, very important, even though originally we think about the genes and proteins. It has been shown that most DNA actually expressed as RNA, and most of them we don't know their functionality. Okay. And a small group of RNA, small type of RNA is called microRNA. Okay. You might have heard about RNA interference. Okay. So people want to know what are those microRNA, how those microRNA actually regulate genes. They want to find the target, for example. And what we did is we have some data from about microRNA, and also we look at the gene expressions of their target, and we want to combine these two data sets to see whether we can have a better understanding which microRNA actually control which gene. Finally, another project. This is what I did during the summer at San Diego. So basically, we know we want to predict the relationship between genotypes and phenotypes. But in between, there are many other things going on. How can we actually take advantage of all those data available? Okay. So what we try to do is given some genetic study, the genotypes and the phenotypes. What we want to do is really to take into consideration all the other information that are available to make this prediction is more consistent, it's more reliable. For example, we do association test. We're going to have a candidate list to show the significance. Whether they are really meaningful functionally, we still don't know. Then let's take a look at those candidate lists to see how those genes expressed in some human tissues that are related to this phenotype. Whether that gives us some more information about their candidate list. Not only expression, but also we have some knowledge about the protein-protein interaction in general in human. If those genes interact genetically to determine this phenotype, you might be able to observe those interactions at the physical level. Whether that information might give us some hints to tackle the gene interaction problem. Also, we have some knowledge about the pathway, disease pathway. You already know some genes involve some disease in a particular way. Then if you have some candidate gene, whether we can rank those candidate genes based on their relationship with other genes in the pathway. So what we did is essentially for candidate release genes, we generate some features based on each of these evidence, and to see whether those features will give us more accurate, say, classification of the disease. Okay, it's almost time. I have to say acknowledgement for my students and my collaborators for all those works, as well as the funding source for this projects. Thank you very much.
Questions? There are no questions? Okay. Yes, Kevin.
You talk a lot about association studies, but I believe there's still a lot of information in families, right?
Yes.
You can reduce noise or the problem of noisy data by looking at families. So,
Definitely.
do you see a lot of your research going back to family studies?
Yes. For example, on the HapMap project, we are currently looking at whether we can actually incorporate the family information into that project. Let's see. Yeah. There are some issues over here because family, they are correlated. How you tackle this relationship. We find some ways. Basically, we want to first take into consideration their relationship before define the phenotype. Okay, we basically first take into consideration their, say we can use some covariance, use the family structure as covariance, and first remove that relationship before we apply these things to them. Yeah, that's one of the directions we are currently looking at.
I have a question.
Yes.
This might be a very naive question.
No.
You said that haplotypes are more informative than single SNP studies,
Right.
which is very logical. But then what determines the length of the haplotypes? Because, you know, there must be a trade-off.
Yes.
So what is the length of width of these haplotypes?
That's actually a great question. That's exactly what we try to do in this model. Because in general, when you talk about a haplotype in a population, you are not talking about the whole haplotype across a genome. Therefore, what we want to do is try to detect those kind of block structure.
And then focus on the blocks.
Yes, each node over here actually represents a couple of SNPs that forms a haplotype segment. And then we generate another haplotype segment. Yeah. Sorry, I didn't have all the details. Yes.
So, as I understand, for personal genomics, for SNP-based sequencing, only to identify SNPs, we focus on common variations, right?
Yes,
this is
that's
the
right.
definition of a SNP anyway.
That's right.
But the variations that are not common might be more serious. That's why they are not conserved. Is there a smart way of identifying those individually without having to sequence the entire genome? Can you think of anything like that?
That's a really hard question.
Nobel Prize, right?
Yeah, you think about it. You are talking about something you don't know, and you don't know who has it, and you want to find it from somebody. It's really hard. I think, yeah.
There is no statistical study that shows the possible locations of.
No, I don't think so. In some cases, a haplotype might mimic the behavior of some rare SNPs, but you still don't know. Unless you know that rare SNP, you still don't know whether it is the case. Yeah. That's why when you have the genome of each individual, definitely you're going to have all the information about all the variation. Yeah, and then what are you going to do? Yeah.
There's no more questions. Thank you, Jing.
Thank you very much. Thank you. Actually, oh, thanks. Thanks a lot.